they do not analyze further in terms of the uh, functional aspects, in terms of 3D structure, and also in terms of protein interaction network. Actually, we can leverage these publicly available tools so that our study could be better, so that we can enhance our knowledge, so that we can understand better about diseases, environmental changes in others. So that's why we always couple this bioinformatic analysis with proteomics. We perform proteomic experiment first, then we continue with bioinformatics. So that our experiment, our study becomes more comprehensive. Okay, for this chapter, we have three learning outcomes. Number one, need to understand. You can understand the database mining process in proteomics. Learning outcome number two, you will be able to identify useful biological information in public bioinformatic tools from existing bioinformatic databases. We can identify the information that researchers need, the information that we can use to solve certain biological problems, and also the information that may explain different experiments. We can combine proteomics with genomic experiment. We can combine proteomic with um, transcriptomic experiment, and so on. Learning outcome number three, will be able to understand how proteomic dataset is produced. So this is about how the information provided in public bioinformatic databases are generated by uh, mass spectrometric analysis. So that's why, as I mentioned just now, after proteomic experiment, after protein identification, we need to channel this information into relevant platform. For example, Uniport database, where almost all biological informations of living cells are stored and managed, so that other researchers or newcomers can learn something from this knowledge database. Okay, so bioinformatics actually bioinformatic database also contains genomic data apart from proteomic data, but the one to be focused in this chapter is protein information only. We focus bioinformatics in the context of proteomics, not genomics. So talking about the protein information in these public tools, public database tools, we need to understand the proteins themselves. We need to know the basic unit that compose a large protein molecules. We need to understand how complex the protein is. And of course, this chapter is related to other courses like biochemistry, where you learn about different categories of amino acids, different levels of protein molecules, and what can proteins do in biological system. So I hope everyone can still remember. It's okay. We still have time to have a look about this fundamental information. especially classification of amino acids and also protein levels. Nowadays, we have 20 amino acids, but in the future, maybe next 10 years, we do not know. The number of amino acids can uh, increase. It could be 25 different amino residues. We do not know because scientific field keeps evolving, and this field always generate new information. As for now, we have 20 amino acids that can be classified into four different groups. One non-polar group and three polar groups. Three polar groups are neutral, positive charge, and also negative charge. Negative charge group also refers to acidic amino acid. Positive charge group refers to basic amino acids. We can classify them into four different groups based on their side chain. Okay, examples of amino acids for non-polar include leucine, isoleucine, methionine, tryptophan, and phenylalanine. Just look at their side chain. Okay, their side chains have only carbon and hydrogen. And in rare case, tryptophan contains nitrogen, but it is in um, aromatic structure. Okay, examples of acidic amino acid are aspartic and glutamic. So far, we have only two acidic amino acids. Just look at their side chains. Their side chains contain oxygen element. Both aspartic and glutamic side chain contain oxygen element, making them electronegative and able to react with other molecules. Examples of basic amino acids are lysine, arginine, and histidine. Their structure, their side chain structure contains nitrogen element. It is another electronegative element that makes this basic amino acid able to react with other molecule during catalysis or chemical reaction. Polar amino acids are serine, threonine, tyrosine, asparagine, and glutamine. Similarly, the side chain of these polar amino acids contain either oxygen or nitrogen element, making them reactive, able to interact with other molecules. Okay, so based on this classification, we can make a conclusion that if the side chain contains um, either oxygen or nitrogen element, they are polar amino acids. They are able to react. Okay, because both oxygen and nitrogen elements, they are electronegative element. They can accept electron from others. This could be seen in the chemical table, which is common to us. On the left, we can see uh, electropositive elements such as hydrogen and nitrogen, where they tend to donate electron. On the right table, chemical table, there are elements such as oxygen and nitrogen, which tend to accept electron from others, so they are electronegative elements. So this is how we compare between non-polar and polar amino acids. It is important for us to understand this because during bioinformatic analysis, for example, multiple sequence alignment, this classification influence the score value we obtain from bioinformatic analysis and also BLAST P analysis. Okay, now we want to compare between neutral polar amino acid and positive or negative charge amino acids. Okay, if the side chain is able to donate or accept a proton, they are classified into these categories: acidic amino acid, acidic amino acid, and also basic amino acid. For the side chain of amino acid, having nitrogen and oxygen element but unable to donate or accept proton, the side chain or its amino acid is classified as polar but neutral. Okay, so we can classify amino acids into these four categories. Number one is non-polar. Number two is polar and neutral. Number three, polar and basic. Number four is polar and acidic amino acids. Four categories we can use to differentiate among them. Okay, is that clear about how we can classify amino acids? Class, is that clear?
Yes.
Okay, moving on to the protein levels. Four protein levels, starting with primary level, followed by secondary, tertiary, and quaternary. Everyone should be familiar with these terminologies: primary, secondary, tertiary, and quaternary. And certainly, quaternary level is the most complex one. Primary level of protein is characterized by linear form of amino acid chain. It is linear, but sometimes it could be like this due to the surrounding area. Okay, no chemical bonds are formed between these uh, amino acids. There is only one type of chemical bond, namely peptide bond. No hydrogen bond, no disulfide bridge, and so on. That's why it is simple and classified as primary. Primary structure could be observed right after protein synthesis, after translation mechanism. When this polypeptide chain or um, primary level becomes more complex due to the formation of chemical bond, as I mentioned just now, hydrogen bond, disulfide bridge, and other type of chemical bonds, they change the shape to alpha helix or beta sheets. Alpha helix and beta sheets are common secondary structure. The polypeptide chain becomes like this due to the formation of chemical bonds. Chemical bonds make the polypeptide chain is arranged in this way. either in helical form or zigzag form. Alpha helix and beta sheets, they are common motifs found in the secondary level of protein. This arrangement of amino acids happen in 3D manner. So that's why alpha helix produced, beta sheet is produced. It still happen in the same polypeptide chain. When two or more, when arrangement of amino acids happen globally, it happens in all parts of polypeptide chain, then the level is changed to tertiary structure. Okay, more chemical bonds are formed, so that's why polypeptide chain usually becomes globular due to many chemical bonds are formed. So we can compare between secondary and tertiary based on the complexity, based on the number of chemical bonds, and also based on the coverage of 3D arrangement of amino acids. In secondary level, 3D arrangement of amino acids happen locally. Only one or two area of polypeptide chains involved in 3D arrangement of amino acids. While in tertiary level, all area of protein chains are involved in 3D arrangement of amino acids, and more chemical bonds are formed. That's why usually we can observe using special equipments, special lab equipments, we can observe protein chains in globular form due to the global arrangement of amino acids and also the formation of many chemical bonds. At tertiary level, we can still observe beta sheets and alpha helix because combination between them is crucial in maintaining the structure of protein at this level. Again, this global arrangement of amino acids and formation of globular form happens in one polypeptide chain. If more than one polypeptide chains are involved, the protein level change to quaternary, the highest level, the most complex one. So the number of polypeptide chains will determine the number of protein domains. For example, hemoglobin proteins in our blood cells. Hemoglobin, it has four domains because it has four different polypeptides that are interacting to each other. And each polypeptide chain undergoes 3D arrangement of amino acids globally. If we want to compare between all these levels, we can highlight the complexity of each level, uh, type of functional motifs that are formed, such as alpha helix and beta sheets. We can compare in terms of the number of polypeptide chains. So this will make comparison more comprehensive and informative. Complexity of protein levels increase from left to right, from primary to quaternary. Only one polypeptide chain involved in primary, secondary, and tertiary level. More than one polypeptide chain involved in quaternary level. In bioinformatic analysis, We can study quaternary, tertiary, secondary, and also primary level. All protein levels can be studied using bioinformatic tools. I think this is the most common protein database we can use as a molecular biologist or biochemist. There might be other more popular databases, but at least we should start with the one that is common and easy to access. It is UniProt. So everyone can access this UniProt database either using a laptop, tablet, smartphone, and others, and it is very to access. And I believe all regions can allow us to access this database. You tinggal di Kuala Lumpur, di Sabah Sarawak, di Kelantan, Terengganu, Johor, semua boleh access. Sebab dia punya server adalah senang untuk di-access dan dia punya performance acceptable to us. All researchers can access this database. Uniport. In this Uniport database, we can see the classification of information in terms of proteins, species proteome, classified proteins, and also the basic informations related to linear protein sequence. Also, the informations deposited in this database classified into uh, manually annotated. So we classified into uh, Swiss spot and also Tremble. Swiss spot contains protein information that are fully annotated, established information, while Tremble database contains protein information that are uh, predicted or computational analyzed, and most of them doesn't have sufficient experimental evidence. We can know the protein function of protein A, for example, in Tremble database, from based on computational analysis, based on comparison with other fully annotated proteins only. But um, the protein A deposited in Tremble database may not have sufficient experimental evidence, but it is still can be used for study by molecular biologists. Okay, that is the difference between Swiss spot and Tremble. Swiss spot contains established proteins or fully annotated protein information, while Tremble contains content information which are analyzed but not experimentally evident, not yet. So any researchers interested to study further, they can use the information from Tremble, and can do experimental validation. Okay, this is another server that molecular biologists commonly use to perform various analyses. It is called ecstasy. Uh, as you can see here, there are many subsections. For example, genes and genome. Number two, proteins and proteomes. Number three, evolution and phylogeny, and others. And In each subsection, there are available tools that we can use either to study um, uh, what composition uh, of protein sequence, or we want to predict the 3D structure, or we want to study protein interaction. So all related bioinformatic tools are available in this protein server, available in Xpeci. At the same time, Xpeci is also connected to other databases. Xpeci is connected to Uniport, connected to ProSite database, connected to Swiss 2D page, and others. So we can see the integration of different bioinformatic tools in one screen. Okay, if you wish expertise, then you can also access other tools huh? Via this one. Okay, moving on to proteomics section of expertise. Here we can see numerous tools such as string, which is for protein interaction study, Swiss model tools for protein structure prediction, foresight for prediction of protein domains and motifs, and so on. So just click any of these link, then we will be channeled to the relevant tool for our study. Mm, right. Oh, okay. Now we go back to Uniport database. Okay, just now I mentioned that the information, this database is classified into two, Swisspot and Tremble. Swisspot contain protein information that are manually, experimentally annotated. Experimentally annotated means we perform the experiment, then we provide relevant experimental information. We provide relevant experimental evidence to that particular protein. So that is why this is called manually annotated, experimentally evident. Annotation process means we add biological information to certain proteins. Another category is tremble, Swiss spot tremble. It contains protein information, but majority of them are not experimentally proven. So the information is based on the comparison with protein information in Swisspot database. Even though Tremble contains uh, half-cooked protein information, we can consider it as half-cooked protein information due to the lack of experimental evidence. Researchers can still use the information in Tremble, can study, and also even publish in journals. Okay, moving on to Uniport, KB, Swisspot, and here we can see too many protein informations that are reviewed or manually annotated. We can classify the protein sequences according to species, such as human, mouse, rat, and also microorganisms. We can see a very long list of protein sequences. There could be uncharacterized proteins. There could be known protein sequences, and all of them are equipped with um, a permanent ID. uh, so those permanent IDs could be seen in this column, entry column. And you will find that this ID is specific and not overlap with other protein sequences. Okay, for example, we click one of them, then we can see all these informations. This information for each ID could be seen in different categories such as function, taxonomy, subcellular location, functional motifs, protein-protein interaction, and many more. Okay, including PTM, standing for uh, yeah. What does it stand for? PTM? Any volunteers?
Post-transcriptional modification.
Okay, post-translational modification. Okay, so this kind of information is deposited in Uniprot database. Okay, we can see the information related to PTM. So by clicking PTM section, we can see the modified amino acid regions. We can see specific specific residues involved, and we can see different chemical processes involved. For example, acetylation, phosphorylation, and also conjugation. And if we want to study further, we can click the link provided here, and we will be channeled to different databases. Okay, moving on to interaction. Interaction. Okay, interaction here on the left panel, we can see interaction. So when we click this part, we will be channeled to different page of information where we could see certain protein interacting with others based on string database application. Okay, so string information can be obtained from this page, and this is what we can observe in string analysis. Everyone could find the full name for string, string. Okay, and it means um, insights into protein-protein interaction network. In protein-protein interaction network, we can see nodes representing individual proteins. The nodes ataupun spherical shape ni, dia refers to individual proteins. And certainly more than one proteins involved here. It could be 10, 20, 100, even 1,000 individual proteins involved. It depends on the level, it depends on the available information in this database. And usually, different individual proteins are distinguished based on their color. Their colors could be light blue, dark blue, light green, and so on. Also, we can see colorful lines. They actually represent different functional interactions. Okay, functional interactions means the possible cooperation, the possible communication, the possible interaction between proteins based on different algorithm. Yeah. For example, based on, as you look at the right bottom right bottom part of this slide, there are text mining, co-expression, and protein homology. So these are the classification of functional interactions between individual proteins. That's why their colors are different. There are green colors, pink, yellow color. Different colors represent different functional interactions. So one individual protein can be connected to one, two, or five, up to 20 individual proteins in this network. So the more colorful lines, the more complex this network. It means that the cellular processes in certain organism is very active and um, important for various purposes. So how to get this? Simply we can use protein ID, or we can use um, protein sequence. Then we will be channeled to different page. Then we can obtain this kind of information. We can download this protein interaction network in one of the buttons here. Okay. Sometimes students tend to make this easier. So students just copy and paste this network in their assignments. Also possible, it's up to you. In one of the aspect assignment will be given later, so you need to include this string analysis. So you can download this image, or you can also copy and paste in your assignments. Okay, gene neighborhood, gene fusion, gene co-expression, and also protein homology. They are all used in string analysis. They are used to classify the functional interactions between individual proteins in the protein network. Uh, Gene neighborhood should, gene fusion should show different colors, different colorful lines, uh, uh, different from co-expression and protein homology. So when we combine these, all information can be integrated, then we can get a comprehensive view about protein-protein interaction network for each protein we use in this string analysis. Okay, for example, gene fusion. It means that, for example, gene A, uh, actually they can separately express. They can actually produce the product separately. Gene A produce protein A, gene B produce protein B. Okay. In gene fusion situation, the genes are combined and their product is also combined. It is called fuse protein, fusion protein. It may happen in certain condition, but we won't discuss further about this. So gene fusion represents gene fusion indicates the protein-protein interaction, and it is used in string analysis. Okay, protein homology. It is based on the information provided by linear sequence of amino acids For example, we have one protein sequence from mouse, then we compare with that from human, chimpanzee, gorilla, cow, cat, and others. From this multiple sequence alignment, we can see the region where amino acids are fully conserved, as shown by V letter, L letter, F letter, and others. So fully conserved region of amino acids indicate the same function of protein, even though they come from different species or organism. Okay? If no conserved regions in this alignment, meaning that the protein sequence from mouse may not function like others. Use this alignment. Okay, moving on to the BLAST-P program. It is about sequence similarity search. I believe you have learned or you will learn about this BLAST in other course for BLAST N program or other kind of BLAST to study for studying nucleotide sequence. Okay, but in SPC server we focus on BLAST-P program for protein sequence similarity search. We need to provide the sequence, usually in FASTA format. FASTA format is characterized by this symbol, larger than symbol, followed by any labels or protein names. And below, we can see the complete sequence of amino acids. Usually, amino acid sequence starts with M, which is 4-methionine amino acid, okay, followed by other residues. We paste this FASTA protein sequence, and we click Run BLAST button. A possible result we can obtain from BLAST-P analysis using SPC server. If we use different platforms such as NCBI, we get a different view of our result for BLAST-P analysis. These are information shown in this BLAST-P analysis: protein ID, length of amino acids, protein description, score values, and E values. Okay, in your opinion, for a significant BLAST-P output, should this value be higher or lower? What if the E value is very high? What if the E value is 0 in this analysis? Any idea about this? Amir Imran. Hello, Amir Imran. Okay, if the E value for any BLAST-P result is high, does it mean significant or not significant? For example, the e-value for BLASP result is 0.5. So is this value significant or not significant for the analysis? Okay, we can go back to the slide. On the right side, we can see a column for e-value for each amino acids. So the e-value here is very low. But my question is, what if the BLASP result is shown by high e-value such as 0.5? Significant. Muhammad Azim replies that 0.5 e-value is significant. Okay, what about Amir Imran? Significant too. Okay, the answer is that high e-value is not significant. Uh, so compared to this one, uh, 0.5 is very high, indicating that the similarity observed may not be reliable. We cannot use that kind of information. But the e-values shown here, uh, these values are very low and close to zero, making this analysis significant. It means that the lower E value, the more significant BLASP output. The higher E value, the less significant BLASP output. For any BLASP analysis, we need to ensure that the E value is very, very low and close to zero. If you get zero E value here, meaning that your analysis is the most significant and absolute. You can use that kind of information for further analysis. So what does it mean by significant result here? Meaning that when you compare your query sequence with uh, the sequence shown here with zero E value, most probably your query sequence is similar or functional like the one listed here with zero E value. Some of you may ask about the query sequence. A query sequence is the amino acid sequence that you use, that you paste in the search box here. Okay, we go back to previous slide. Example of protein shown here is UDP galactose phosphate transferase. So this is what we call query sequence. Query sequence could be any proteins from human, bacteria, plant, and so on. Then we paste the sequence in this search box. So this is what we call query sequence. Okay, when query sequence is compared to other proteins and the E value is zero or very low like this, meaning that the BLASP result is significant and you can rely on this result. You can use this result for other analysis. Okay, as I mentioned just now, the lower E value, the more significant BLASP result. Okay, can also perform BLASP analysis using another platform, namely NCBI. It is not available in Expertise server, so we need to search for it. We need to use Google search and type its name, BLASP. Okay, front page of BLASP platform in NCBI. Similarly, we need to paste our query sequence, any protein sequence here in this chat, in this search box. There are also other parameters that we can control. It depends on us. If we want to ignore, it's fine, then we can proceed. Some researchers want to compare the query sequence in this search box with those in certain organism. So that organism names could be typed here in this search box. Okay. For example, Homo sapiens for human, Pseudomonas aeruginosa for bacterium. Just type the name of organism, meaning that your BLASP analysis will compare between your query sequence with proteins in the type organism, not other organism. If you want to compare your query sequence with proteins from all types of organism, so no need to mention the name of organism here. Need to skip this one. Okay, the result view from BLASP analysis using NCBI slightly different from what we see in Expertise server. Here we can see max score value, total score value, coverage, as well as percentage identity. And I believe everyone could understand that the higher percentage identity, the more significant our BLASP result. Percentage identity. If it is 100%, meaning that our query sequence is identical to the number one here. Okay. And usually 100% identity is coupled to zero e value, indicating significant result. Okay. If lower protein percentage identity observed, meaning that uh, our protein sequence is similar to them but not identical. Okay. Sama tapi tak serupa. That is the difference between similar and identical. Similarity and identity. Okay, so far any question? Any question? Siti Aisyah Yusof. Any question?
Not yet. Better.
Okay, I have a question for you, Siti Aisyah. What if a BLASP result shows lower percentage and higher e value? What does it mean? Lower percentage identity and higher e value. What does it mean?
A higher E value means the it's less significant.
So, what can we conclude about our query sequence? If the percentage value is low and E value is high,
It's not identical.
it is not identical. Okay, meaning that our query sequence is not comparable, it's not similar to deposited protein sequences.
Yes.
Okay, thank you. Um, we continue with the following slide. Okay. To summarize, these are uh, important terminologies in BLASP analysis. Pairwise, multiple sequence, alignment, PAM, blossom, evalue, at least. At least you can differentiate between them. E-value, blossom, PAM, pairwise sequence alignment, multiple sequence alignment. Pairwise means you compare between two protein sequences, pairwise, only two protein sequences. Multiple sequence alignment, it is about the analysis involving more than two protein sequences. We can use 100 protein sequences in one multiple sequence analysis in order to identify the conserved regions. Usually, we perform MSA or multiple sequence alignment because we want to identify the amino acid regions that are shared with other protein sequence from different organisms, for example. We want to identify the conserved amino acid regions in the sequence. That's why we compare with more than two protein sequences. For example, we compare the sequence of proteins from human, rat, cow, snake, and others so that we can identify the shared amino acid region, the conserved amino acid region. This is important because we want to understand the function of protein sequence. If we compare between only two protein sequences, usually we want to determine the identity of query sequence. Our query sequence usually is unknown. For example, we obtain this unknown protein sequence from mass spectrometry analysis. We do not know the name of this protein function, the name of this protein sequence. We do not know the function, structure, and so on. So that's why, as the first step, we compare the unknown protein sequence with protein sequences deposited in databases. So this is called pairwise sequence alignment. Okay, PAM and Blossom, they are platform they are metrics used to carry out multiple sequence alignment. Usually, we choose PAM to study closely related protein sequences. For example, protein sequence from Salmonella type murium and also protein sequence from Salmonella type. They are closely related. They are from the same species. Uh, but we still want to identify the conserved region. So we use this matrix, PAM. If researchers interested to compare a protein sequence from Salmonella with protein sequence from Antarctica bacteria. Ah, yang kita tak pernah jumpa kan. Tiba-tiba ada. Okay. So we want to compare between them. Known Salmonella species and unknown Antarctica bacteria species. And we can use Blossom platform. A substitution matrix to compare between evolutionarily divergent protein sequences. The last one is value. In simple words, E value is about the noise or background value. Noise or background value is not wanted at all. If possible, we can completely eliminate the noise or background value. So that's why, as I mentioned just now, the lower E value, the more significant our result. And if E value is 0, meaning that our result, our BLAST result is most significant. E value may vary across different protein sequences in the list. Okay, we go back to previous slide. Um, okay, here. Hmm. E value for each BLAST output varies across different protein sequences. For example, protein sequence number 1 and number 5. Both are listed and both are significant, but their E value is different. Okay? E value for number 1 compared with E value for protein sequence number 5. So which one is more significant? Certainly the protein sequence number 1, because its E value is lower than that of protein sequence number 5. In other words, the most significant result is rank number 1. Dalam protein list yang appear dalam BLAST analysis, yang paling atas itu adalah yang paling significant, paling penting lah. So the rest you can ignore. But if you want to look the similarity with those from other species, yes, you can still list the top 10 or top 20 protein sequences as long as their E value closer to 0. Um, okay, so that is about E value, and the threshold is shown here, 1 exponent 6. So this is the threshold value. Higher than this may indicate insignificant BLAST-P result. Lower than this threshold indicate significant BLAST-P result. Okay. The lower E value, the more significant our result. Okay, here we have 4 examples, A, B, C, and D. So in your opinion, which one is smallest? Which one is most significant? A or B or C or D? Any volunteers? A or B or C or D? Nur Alia Zulaikha. Any  answer from you?
A. A.
'Okay? 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 'Okay Are you here, No Alia Zulaika? Tak ada respon. Tak ada respon ni meaning you may be absent lah kot. Kan? Nur Izzah Hana? Are you here, Nur Izzah Hana? Okay, Nur Izzah Hana replies B. Okay, apa kata Nurul Almas Yusaini? A or B or C or D? Which one is most significant E value?
Um, B juga kot.
B. Ni ikut kawan ke ataupun faham?
B atau D?
B. Jawapan awak B
Mhm.
Okay. Lain-lain. Syafi Hilmi Zainal. A or B or C or D? The most significant E value from blast P? Mana yang paling signifikan ataupun yang paling kecil lah noise value?
Oh, yang paling kecil. C lah paling kecil.
So itulah jawapannya, paling kecil. Thank you. So the answer is C, the lowest E value. The most significant blast P result. So as you compare between them, their decimal points do indicate their whether each value closer to 0 or closer to other numbers. So yang paling dekat dengan kosong adalah C lah. 7 kali kan? 1, 2, 3, 4, 5, 6, 7. Okay, we continue with metrics that we can use to study the conserved region during multiple sequence alignment. PAM and BLOSSOM. As I mentioned just now, PAM is for closely related protein sequences, BLOSSOM for divergent protein sequences. Okay. So there are values in this matrix ranging from -4 until +3, I think, for PAM. And ranging from -7 until +17 in PAM. While in BLOSSOM matrix ranging from -4 to +15. Of course, the values are slightly different from when we compare between PAM and Blossom matrix. Then from this matrix, actually we can do classification. For example, Blossom matrix, we can see the top region represents small and polar amino residues. That's why just now I mentioned we need to understand the difference between polar and non-polar, basic, acidic, and also neutral. Because it is used, that principle is used in this production of matrix, yeah? MSA matrix. At the bottom, we can see aromatic amino acids. So from this blossom matrix, we can see different categories. Okay, now we want to understand about how to calculate the score value for each BLASP analysis. Okay, for example, we want to compare between two sequences shown here, NDEQ versus NTWQ. Okay, we start from the first one, NDEQ. On the left, we can see the N letter, N residue. We drag to the center of this matrix. Then it should match another N letter in horizontal line. So the value is 6 plus 6. For the second residue, D, on the vertical, we need to find the match value with T. So the value shown here is -1. We do the same thing until all pairs of amino residue can give us score value. In total, we can get the net value for this comparison of protein sequences. Okay. What if we compare the identical protein sequence, NDEQ number 1 and also NDEQ number 2? Similarly, we do this starting with the first amino residue on vertical, we drag to the matrix. We can find the value 6. Second residue D, we drag to the matrix. And the value is 6. Not in the red circle. D, D. And the score value is high in total. That's why identical amino sequence, they can produce higher score value. While similar but not identical, they produce lower score value. In other words, the higher score value, the higher percentage identity. The lower score value, the lower percentage identity. Is that clear? How to calculate the score value? Okay, another example is comparing between these sequences, AARR and WTT versus AARD and YSS. 8 amino residues for protein number 1, also 8 residues for protein number 2. We compare between them. Out of 8, 4 are identical, namely residue A, R, and N. Out of 8, 7 similar amino residues. Okay, so the value is different here. Identical amino residue mean same, exactly same residue. Similar amino acid means same nature only. It could be different amino acid residues, but in the same categories, same nature. So usually, the value for similarity is slightly higher than the value for identity. Okay, in pairwise sequence alignment, we can see this information: mesh, mismatch, and gap. Mesh refers to identical amino residues in this comparison between two sequences or 3 or 5 or 10 protein sequences. Mismatch mean different amino residues. It could be from different categories of amino acids. Gap, you can see uh, incomplete pairing process, incomplete comparison. If we compare between two sequences, only one of them having amino residues, another one no residues present here. This happen when the bioinformatic tools rearrange the sequence so that they can be aligned. If we do not do alignment, we could not see this gap. But when we perform alignment, all amino residues are arranged so that we can identify the conserved regions. For any position having no residues, for example like this, it shows a gap here. So ini boleh lihat dalam BLASTP result. You need to scroll down the BLASTP result and you will see this. Mesh, mismatch, and gap. Okay, I think that's all for today. Next week, we will continue with this chapter, and uh, we will have uh, online lab session for another lab activity. So that you can we can discuss about how to analyze the protein sequence and how to perform protein structure prediction next week. Okay, that's all for today. Thank you. Wassalamualaikum.
Thank you.
Thank you, Doctor.
Thank you, Doctor.
Salam. Thank you, Doktor.